Welcome to This Explainer. Today, we're diving straight into a massive problem in modern oncology. We're going to push way past the standard limits of how we currently understand tumor progression, and we're going to ask some genuinely hard questions about the philosophical and biological frameworks guiding cancer research. So let's just jump right in.
For decades now, the biomedical community has leaned heavily on these incredibly powerful, yet mostly descriptive, frameworks to study cancer. I mean, we can sequence genomes, we can map out cellular structures, and we can pinpoint literally thousands of associated biomarkers. But here's the absolute kicker. Do these observations actually tell us why a tumor progresses? Or are they just showing us how it looks at any given moment? Basically, are we confusing mere correlation with actual causation inside the hyper-complex tumor microenvironment?
Let's explore that in Section 1, Beyond the Hallmarks.
We really need to start by looking critically at the whole conceptual foundation of modern oncology. Historically, the field's been philosophically split between two major camps. On one side, you've got the cell-centric somatic mutation theory, or SMT, which essentially argues that genetic mutation is the primary initial spark, the root cause of cancer. On the flip side, there's the environment-centric tissue organization field theory, or TOFT, arguing that the real root cause is a disturbance in the surrounding tissue architecture. You know, it's the classic seed versus soil debate. But as we're increasingly realizing, the reality is a deeply integrated, messy combination of both genetic and microenvironmental factors. And that brings us straight to the famous hallmarks of cancer, established by Hanahan and Weinberg. Now look, it's a brilliant synthesis, no doubt about it, but we really need to consider its heuristic limits. Philosophically speaking, the hallmarks are largely descriptive. They lack true spatiotemporal integration. And honestly, the biggest issue is that they often confuse facilitating conditions with actual driving causes. It's like they tell us what traits a cancer acquires, say dodging the immune system, but they entirely lack the underlying mechanistic explanatory power to tell us exactly how that causal chain is actually built. So how exactly do we fix this? Well, welcome to Section 2, Philosophy Meets Oncology. If we're going to move beyond just observing things and actually close this explanatory gap, we have to start integrating formal philosophical theories of causality right into our biomedical research. Let's kick this off with a concept known as the astonished condition. And hey, don't let the acronym intimidate you. In the chaotic, hypercomplex interactome of the tumor microenvironment, singular isolated causes almost never exist. It's just not how it works. Instead, we have what philosophers call a causal pie. An astonished condition just means a specific factor might be insufficient all on its own, but it's a necessary slice of a much larger pie of conditions that, when put together, are sufficient to actually cause that tumor to progress. To really elevate our thinking here, we should look at Judah Pearl's ladder of causation. It's a fantastic mental model. Down at the very bottom rung, we have associations. This is merely seeing that two things happen to occur together. And honestly, a huge chunk of current cancer research is completely stuck on this bottom rung. To really understand causality, we've got to climb that ladder. We need to move up to interventions, actually doing something, like actively blocking a pathway, and seeing what happens. And ultimately, we absolutely must reach the top, counterfactuals. That's where we imagine what would have happened to a specific patient if we had intervened differently. To make that climb, we're relying on some cutting-edge modern causal frameworks. By bringing in James Woodward's interventionist theory, Judah Pearl's probabilistic directed acyclic graphs, which we usually just call DAGs, and Nancy Cartwright's intense focus on single-case clinical realities, we can actually begin to mathematically formalize the absolute chaos of the tumor microenvironment. We can literally map out these variables as a functioning network and calculate the actual flow of causality. So the crucial takeaway here is perfectly summarized in the Russo-Williamson thesis. It sets an incredibly rigorous standard. If you want to make a true causal inference, you absolutely cannot just point to a statistical correlation. You have to also prove the underlying mechanistic explanation. It's only when you combine both of those elements that you can establish a definitive, bulletproof causal claim in tumor biology. Okay, let's see how all this philosophy translates into the real world. In Section 3, Prototype 1, Diffuse Gliomas. Let's anchor these high-level philosophical concepts straight into hard biological reality by analyzing our first multi-scale model. Gliomas give us a pretty perfect test case for this. When you contrast IDH wild-type glioblastomas with IDH mutant gliomas, you're looking at entirely distinct causal topologies. The lethal wild-type is aggressively driven by these rapid cycles of invasion and compaction, paired with really high angiogenesis. The mutant type, on the other hand,
It's got a prolonged dormant phase and very weak angiogenesis. Because of that, they require completely different causal maps if we ever want to truly understand their progression.
And the incredible thing is, we can map them mathematically using structural equation modeling. By utilizing these algorithms, we're moving light years beyond basic descriptive pathology. We are essentially mathematically formalizing the dynamic feedback loops between things like oxidative cells, cellular proliferation, tissue compaction, and tumor jamming. We're literally coding the causal network of the cancer. So, what exactly happens when we do this? Well, we find the true drivers. For instance, in glioblastomas, causal modeling successfully isolated the interaction between TSP1 and CD47, working through the TGF-beta/SMAD3 pathway. This isn't just some randomly correlated biomarker that happens to be hanging around. The math actually proves it is a definitive, mechanistic, causal driver of tumor invasion. That is huge. And when we look at the slower-moving IDH mutant gliomas, we take a massive multimodal approach. As this research lays out, if you feed vast amounts of data, we're talking everything from gene expression transcriptomics to protein levels and even radiomic imaging directly into these causal frameworks, we can yield highly precise, predictive signatures for exactly when that tumor is going to recur. We're essentially mapping the entire causal journey of the disease.
Now, let's look at another example in Section 4, Prototype 2, renal carcinoma.
Our second prototype is clear cell renal carcinoma, and here the network is remarkably different. It's heavily dominated by an initial genetic inactivation that leads to this stark, almost perfectly linear causal chain. It all kicks off with the inactivation of the VHL gene that directly stabilizes HIF factors, which then activate the IL-34/CSF1R axis. And what does that do? Well, it physically drags macrophages right into the microenvironment, forcing severe immunosuppression. It's literally a domino effect of causality right there in the tissue.
Now, why does charting out this specific causal DAG actually matter? Because when we understand that exact chain, we know exactly where to strike. Applying Woodward's interventionist logic, if we apply a pharmacological blockade right at that IL-34/CSF1R axis, we logically break the chain. We stop the macrophages from migrating in, we reverse that terrible immunosuppression, and boom, we restore the efficacy of our standard treatments. Causality literally gives us the precise target. Which brings us to our final act, Section 5, the future of causality. You really have to ask yourself what happens to the entire field of oncology when we start systematically integrating this formal causal analysis into all biomedical research. Well, it completely revolutionizes how we implement care at the patient's bedside. We transition away from those generic, population-based statistical correlations and move into an era of true precision medicine. We're talking about developing advanced multi-scale organoid models. We can simulate therapeutic counterfactuals in clinical trials before we ever give a human patient a single drug. We'll be mapping our treatments directly to an individual patient's entirely unique causal DAG, which finally allows us to manage the insanely chaotic heterogeneity of these tumors. As the source thesis so powerfully concludes, conquering the hypercomplex interactome of the tumor microenvironment truly demands a holistic marriage of rigorous philosophical logic and empirical biology. The descriptive era, the one we've been in for decades, it brought us incredibly far, but it's this new causal era that's actually going to allow us to truly control the disease. The scientific paradigm is fundamentally shifting right beneath our feet, moving from mere correlation to true causation. We are no longer just casually asking what is inside the tumor. We are fiercely demanding to know why it behaves the way it does. Which leaves us with a really critical question to ponder today. As this new paradigm takes hold, how are you going to redesign your own thinking and your own research to uncover the true mechanisms driving your data? Thank you so much for joining me on this explainer. Keep asking the hard questions, and I'll catch you next time.